to the Weekly Bioanalysis, a Kcas podcast.
Hello, and welcome to the 37th episode of the Weekly Bioanalysis, the official podcast of Kcas Bioanalytical and Biomarker Services. Kcas is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is Dominic Guarino, and I've been in the immunology and gene therapy space for over 26 years. I'm here with my co-host, John Perkins.
Hello, I'm John Perkins. With my focus on LCMS technologies, I've been with the bioanalytical space for a little over 26 years.
John and I are senior scientific advisors at Kcas, and either or both of us are available to answer any questions you may have regarding this podcast or any of Kcas services. We have two very special guests, and I'll let them introduce themselves. One is John Pirro, and the other is Stephanie Pasas-Farmer. John, tell us about yourself.
Well, thank you very much. My name is John Pirro, and I am the Senior Director for the Large Molecule Biopharma LBA group here for regulatory and non-regulatory groups. I joined Kcas in April of last year. I'm very excited. I've had over 15 years in the CRO industry for bioanalysis of large molecules, as well as immunogenicity going forward. Thank you.
And Stephanie?
Hi, I'm really happy to be taking a part of this podcast. I'm Stephanie Pasas-Farmer. I have 17 years of bioanalytical experience in both large and small molecule. I was gainfully employed in the contract industry and quit and started a scientific advisory firm 6 years ago. And then out of that work, we developed Adriadne Software, which is what we're going to be talking about mostly today. It's a human augmented intelligence tool for the review of PK and ADA data. So thank you. I'm happy to be here.
Yeah, thank you both. We are so excited to have you. We are thrilled to be here with our audience. For those of you that are new, our podcast is now available virtually everywhere. Apple Podcast, iTunes, Spotify, Google Podcast, Amazon, and many other outlets as well. Wherever you choose to find and play your podcasts, we would love to have some feedback. So we always appreciate anybody posting feedback. The podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from you all. We're constantly looking for topics, and we'd be happy to discuss something that you may want us to cover in the future. So we're very excited to be here this week. We're looking forward to a very fun episode today. Before we get into the news, John, why don't you give us a little bit of a teaser about what we're going to cover today in podcast number 37?
Sure. As usual, we're going to follow the same structure we always go with. We'll go through the news, looking at COVID topics, non-COVID news, and some investment items too. And then our main topic is what COVID has taught us about limitations of the drug development process. And this is a chance for Dom and I to sit back and let Stephanie and John tell us all about the use of AI and how it's helping our processes.
Great, John. Let's kick us off with the news.
First up is a topic that is actually a follow-on from last week, and we had some discussion about vaccine doses for people who'd previously been infected, and there's actually been multiple studies coming out, mostly unreviewed, but still sort of corroborating the same finding that actually people who've been recently infected with COVID should go ahead with vaccination, but they may not need two shots because what the single shot seems to be enough to sort of turbocharge their immune response and actually build up very quickly.
Yeah, John, this is something that we, got asked about by one of our audience members, and it continues to sort of evolve. But I think it makes a lot of logic, in terms of if you've been infected and you've already got some central immunity, then maybe perhaps you're protected after a single vaccination. Not shocking news here. I think we'll learn more about this over time, but I think even if you have had COVID, you get both shots, you're still safe. That's the message. You're going to be safe, and that's really what it's about, right, John?
Yeah, I think that they did, one study said really for people who've been previously infected, the second booster didn't make a great deal of difference, but it's not going to hurt to have it. It just won't make a great deal of difference to how your body's reacting. So we talked about the Kcas School of Medicine at Mount Sinai and a Baltimore study. This is now a group at the Fred Hutchinson Cancer Research in Seattle and New York University School of Medicine, all coming out with like similar findings. And it was the Fred Hutchinson one is actually very interesting because it was like after the first shot, not only were participants, they could actually they showed good effectiveness against the South African variant, which is of all the ones we talk about the biggest concern, because it seems to be that more evasive in terms of the current vaccines. But because the body has this boost, the people who had been previously infected dealt with it much better.
Yeah, John. It's definitely evolving, and we're continuing to learn about it, and excited to continue to monitor it over time. But John, sounds like we got a jam-packed show today. Let's move on to our next
Sure. Yep.
topic.
It's just an interesting topic to start with. So the next topic is actually this is a completely different direction. So there's been sort of rumblings about this in the media, but it's really in terms of the backstory to this is with COVID and limited supply of vaccine, there has been concerns voiced about disproportionate distribution of vaccines in the developed world. So basically, the US, Canada, European nations have all made sure that not only do they have sufficient supply for their population, they in many cases have surplus supply. I know Canada had a huge excess, and we talked a few weeks ago about how New Zealand, in terms of making sure that they had sufficient supply, they were going for excess, but they were committing to distributing to other Pacific Island nations. But this is really talking about in Africa, things are starting to move forward. So there's the WHO initiative COVAX, which is reliant on working with the vaccine producers and their commitment to provide doses. The COVAX is then looking at how can they get the vaccines out to developed nations, and there has been concerns over the past few months about how slowly the COVAX is ramping up, but they actually have moved forward in terms of distributing 600,000 doses to Ghana, and then Cote d'Ivoire is second in line within Africa, and so it's a step forward in the right direction. Africa is way behind where really they need to be in terms of the population on the continent,
Yeah.
but at least it's moving. I'll let you chime in, and
Yeah, I
I'll
mean
see what to say.
it's staggering, right? So in this article, it touches on just how poorly the distribution is, right? So 17% of the world's population lives in Africa, right? But they've gotten just 0.2% of the 209 million doses that are available. So that just right there is it's alarming to read, John, right? And so
Sure.
but there it's not all bad. There are, like you said, the COVAX initiative is really starting to maybe kick in. Look, we've had our own issues here, in the US. That is starting to alleviate itself, and I think we're going to touch on that in some subsequent topics here. But obviously, we've talked significantly about one of the downsides to both the Moderna and the Pfizer vaccines is the cold chain. That becomes an even much much more of a logistical challenge in let's say resource poor nations, right? And
Sure.
but it's great to read about it. It's a fascinating article that we're kind of going over. And last thing I want to touch on is actually looks like the AstraZeneca Oxford vaccine is being the predominant one entering to Ghana, right, John? Do I have that right? And
In
our buddies at the Serum Institute
Ghana,
actually made
yes,
it, right? Yep.
yes, absolutely, yes, and that is an interesting, because South Africa actually has put the AstraZeneca one on hold because they're not convinced about its effectiveness against the B.1.351 variant.
Well,
But you got other ones coming through that we'll touch on later that are showing effectiveness, and I'm sure that will all shift as well. But some countries have actually moved forward with the China produced the
yep.
Sinopharm vaccine.
Hmm.
Others are actually testing the Sputnik 5 vaccine in pilot programs. It's slowly getting there, but really Covax is going to be key. And then that rolls into the
Yeah.
next article where Novavax, who are another of the really promising vaccines, who hope for emergency use authorization in May in the US, they've actually committed 1.1 billion doses to GoVax for low and middle income nations. So there is hope on the horizon. You know, that actually almost effectively doubles the number of doses that have been committed to COVAX so far.
Yeah, and again, this is our friends I call them our friends, John, like I know them, right? But the Serum Institutes of India have made those doses. So there's it's lining up nicely with what we've been sort of, I hope, providing information to our audience around in terms of um, why some of the challenges with what's happening and just the sheer pace of it that's happening. Remember, we're still only maybe 12, 13 months into this. And, we got 1.1 billion doses going to, um, you know, middle income nations is how they term it now.
Sure.
So
Yeah.
it's fascinating. And this is, Novavax is one that was kind of maybe lagging behind a little bit, but obviously has caught up. It looks like their phase 3 trial is quite effective, showing, I think you touched on some of those. I don't know if you gave actual stats, but it looks like it's like 90% and
Yeah. It's
50% in the variant. So all good stuff, right? I mean, this
yeah.
is
It's
fascinating. Yeah, go ahead.
Yeah, we talked about it last week. It's shown to be effective against the UK strain, so it's definitely showing effectiveness. One thing I didn't mention, I think we've been referring to it as an mRNA, which it's not.
Yeah.
It's actually a full-length perfusion spike protein created with recombinant nanoparticle technology, and they have a proprietary adjuvant. So it's a 2-dose regimen, 1 month apart. But again, nice thing about this is it's stable at refrigerated temperatures, so you have that a bit that
Mhm.
makes your logistics that much easier. So
Yeah.
yeah, there's the path to Africa is there. It's just that difficulty of getting it going.
Yeah, and Novavax is obviously a very big player in this space, and you touched on it. Their technology is not just in a protein base, but it's also that adjuvant is really part of the
It,
sort
it's
of
in just quickly,
proprietary intellectual property, and it's great stuff. But maybe
just
we'll
to
do a
quick,
reboot on vaccines over time now that we got so many on the market that are popping
sure.
up. Right, John?
Just a quick aside on Novavax. There was actually an article today that said they almost went out of business, or
Yeah.
they were getting close to going out of business until the COVID came along, and here they are talking 2021 could be a bumper year for the company, and it's totally changed their outlook. So
Yeah,
let's move
I
on.
think it's happened to. Yeah, we can move on, but I think that's happened to a lot of companies, and in our space are really feeling some of that benefit, or at least. And again, let's move on. But there's a lot of great movement in the uh, initiative on COVID, right? So, next article, John, it's insights into how coronavirus changes shape. Could a development of the COVID-19 vaccines. So this is coming out of a couple groups, academic groups, right, John? The Yale and University of California at San Diego, and they look like they're working on changing structures of the SARS-CoV-2 coronavirus, and that's caused all the pandemic and everything. And this actually, I think, is something where I wonder if they got some AI in there. But tell us, it's focused on the ACE2 um, receptor and everything. But tell us a little bit more about how they're doing some of this.
So basically, Yale using imaging techniques to watch how the coronavirus spike protein changes its shape in response to antibodies. They sort of had previously identified two types of spike protein structure: a closed state in which all receptor binder domains are oriented downward, an open state with up to 3 receptor binder domains oriented upward, making them accessible to receptors. And they're saying that if they can keep it in the closed state, then they can
Mm.
prevent infection. It's really that shift, and they noticed. Yeah, I'm trying. Yeah, that's
It
one
leads
thing.
nicely into some of today's guests, right? We don't need them to if they want, but it's about neutralizing antibodies essentially, and that's part of what we'll talk in terms of like in the drug development world, typically neutralizing antibodies are unwanted effects, but in the vaccine space, they're exactly what you're looking for. So, but that's really what this is about when you talk about closed receptor bound and open states. That's the whole principle behind the mechanism of action, right?
Yes. So
Generating
yes,
antibody that binds
yes,
to it.
yes. So some antibodies attach when it's in the open position, competing with ACE2 for binding. So then the virus can't reach human cells. Others attach to the closed space spike, stabilizing the protein, so it can go to the open state, and that's where Yale's postulating this would be a good way to attack in the longer term to actually prevent that ability of the virus to attach.
Yeah,
And
and
then
well, oh, sorry, go ahead.
then the University of California, San Diego are actually looking at the sugars that coat the spike protein structure that actually help it evade the immune system. So it's a slightly different approach, and they actually found it's just one glycan was responsible for the opening of the spike protein. So if you can halt that or interrupt that, then there's your mechanism to
Yeah,
possibly come up with
and,
a
and viruses are tricky,
therapy.
John. They will
Oh,
change their
oh yeah.
glycosylation the patterns. They'll shed things. So that's an interesting that finite. It's wonderful for them to. I bet you it's a fascinating read to see how they came to the conclusion that it's a single end glycan that is the real contributor here. But yeah, no, this is so we'll be learning about this in years to come. I feel like we're going to learn about ACE2 genetics more than we want to, because I feel like that'll be the keys to unlocking why some people are asymptomatic, why some people are maybe
Yeah,
perhaps more susceptible. All of it may lie in there. And then, I think hormones are playing a part in this as well. But I do have to give a plug. One of the collaborators, University of Pittsburgh, my alma mater, they ran some of the algorithms. I knew this was going to be a lot of this is some when I think algorithms, modern day artificial intelligence, or big data type things. So nice there. John, this is maybe we'll move on to really what is the highlight of what's happened in the last couple
sure.
of weeks, right? Why don't you kick us off here with really what has been, I think, some of the biggest news and
Well,
really,
it is the biggest news. Really,
yeah,
we all routinely talk about the Pfizer and the Moderna vaccines. When we hear someone is getting the vaccine, it's always like, well, which one are you getting? Or is
yeah,
it the Pfizer, is it Moderna, or with people in the UK, is it the AstraZeneca? Um, but we actually the Johnson and Johnson vaccine was reviewed by the FDA just last weekend, and so it received, I think, unanimous emergency use authorization, and so vaccines with the J&J started shipping 1st of March. This is a completely different approach from the ones that are already out there and being administered. It uses a viral vector where as means of stimulating the body. But the big deal is it's a single dose
hmm
regimen. So again, if when we're talking, 100 million doses, that's 100 million people. Whereas when we're talking Pfizer and Moderna, when they say 100 million doses, that's only 50 million people. So that
yeah,
ability to get out to much more people in terms of immunization is a huge deal. So I think one thing about J&J, they have been slow in ramping up supply, but Merck are coming on board to help double production of this vaccine. So again, it plays back into the collaboration and
yeah.
cooperation we've been seeing between pharma companies, which is great from our perspective. And yeah, the expectation is 100 million doses by the end of June, but a big deal.
Yeah, and it's refrigeration. It's a protein based, really fascinating kind of dynamics in terms of working with Merck to allow them to manufacture them and help distribute it, and even like I don't know, on the heels of this, our local grocery store is opening up to. It's called Hy-Vee, and you're now eligible to sign up and get a vaccine at Johnson and Johnson. Excuse me, at Hy-Vee locally here in Johnson County. So that I don't know, it coordinated with this, although they are offering a different vaccine. I think you're going to feel more ability to get a vaccine is where I'm going. I think not only will Moderna and Pfizer start to roll out at a faster rate because of Johnson and Johnson impacting all of it becomes a little bit business-like, but benefits, I think, the population quite a bit, and really just seen a massive, in my opinion, surge. Lots of family members on my end are getting vaccinated. We'll probably start to think about getting in line soon. And John, with any bit of luck, maybe by June, July, anybody who wants a vaccine can get one. And that actually leads nicely into our next
So,
topic, right?
so
Which
I
is, yeah, you
I
got
was
one more point on that, and then we'll go to the
yeah,
next one. Go ahead.
one more point. I was actually going to mention this later as we're talking about what's coming up. Actually, the rules in New York have changed. Where it was only frontline workers and over 65s, middle of February, it actually switched to if you they have a list of underlying medical conditions, of which on one front I'm fortunate enough to have, which I'd rather not have in the longer term. But I have got my appointment for my first shot next Wednesday.
Oh, wow.
So yeah. So it was actually one of my friends from my previous job posted on Facebook that she had got her first shot. So I quickly sent a message. Well, like, what was the difference? And she told me, and yeah, I booked my appointment, and there I am.
Yeah,
And
no,
look,
and that,
we've got
there's there's people at KCS that have some similar things, John, and they got vaccinated this week. It's good to hear in your home state of New York. It's a similar vamp up. Just wonderful. There was a little bit of a buzz even around the facility because people were signing up, and some people are driving to Lawrence, which is like 30 minutes away. My wife looked last night. There's no way I'll get anything this week, but I think over
yeah,
time, maybe even by our next podcast, I'm hoping I'm either on the list or vaccinated, but it's good to hear that you are, John. So
yeah.
that's fantastic. And
It's
yes,
it's a
Stephanie,
wor-
you
it's
can
a
absolutely chime in. Go ahead, Stephanie. Tell us your thoughts. Absolutely.
I was just saying that the rules have changed for my California relatives. I'm originally from the West Coast, and I do have a lot of family members. My cousin has a child with Down syndrome, so they're also able to be because they're caregivers for Danny.
Wonderful.
Yeah.
Wonderful.
yeah
So
So
that
caregivers of, is that what I heard you say? Caregivers of,
yes,
oh, wonderful.
yes. So my cousin, yes, her son has Down syndrome. So because she's the primary caregiver for the family, she's able to also get it, so which is nice.
that's great. That's great.
Yeah.
Yeah, so
Um,
it's a 1 hour drive for me to get it. It's at Syracuse, at the State Fairgrounds in Syracuse, but definitely a worthwhile 1 hour drive. So looking
well,
forward to it.
that and thanks for sharing that. Good to hear. And of course, California. My home state's Massachusetts. They followed with starting to open up stadiums, and it's great to hear that they're progressing in California as well. So, all around great stuff, John. And because let's wrap up our news and notes. We got a couple more items, and one is John. We've talked about this a lot, right? Like at one point, it was as low as like under 50% of Americans that they'd get a vaccine, but that has certainly changed now, hasn't it, John?
But
Right? According to
this is more on the farmer's reputation. So
sorry.
when we started the podcast, it was farmer were, I think, the only body that was less trusted than farmer was the government, or viewed positively. But so when the poll is, it's a Harris poll that judges how people view industries, etc. A year ago, or in January 2020, the pharma industry was viewed positively by less than a third of the population. But in the most recent poll, rating on a 7 point scale, 62% people rated pharma as with a 5, 6, or 7, which is obviously judged positively, and the Harris basically attributed to the fact to the how pharma has responded to COVID-19. So
That's
it's all about the quick jumping on quick development of vaccines, that sharing of resources across the industry we talked about, trying to getting speeding vaccines to market that have high efficacy, and yeah, it's all about that in terms of that's changed the public outlook. So
yeah.
it's now the question is, can pharma sustain that positive outlook from the public, or will price concerns take over in the longer
Yeah,
term?
no, John, you kind of, if there was a bubble over my head, you would have just read it, right? Like this is great, but I'm sure if fast forward a year from now, if the hoopla around COVID is hopefully behind us, and we're just routinely getting vaccinated, I'm sure attention will shift back to pharma in a negative way, but let's hope it doesn't. And I bask in it now is what I say
Yep.
to pharma
Yep.
and biotechs. Bask in it now. Congrats, you're over 62% and more favorable in the certainly the government. And I'm guessing you passed the cable company, which I think is like always top 3 and most distrusted or disliked. But John, actually, I maybe for next,
So
let's
can I
kind of
just
research who's taking the vaccine. I misspoke when I started this one, but I wonder what the rate. I felt like I saw something recently that it's over 50% of Americans will now take it. But let's move on, and maybe we'll think about getting a news item to see what the polls are on
there
that.
is definitely demographic gaps in terms of who's willing to
Yeah,
take it and who's not, and it's a question of how you get over that demographic gap to reach out to the people who are at
yeah,
the moment are
some
saying
some
no.
misbeliefs, right?
Yeah,
Um,
exactly.
But John, we're going to wrap up with a couple investment things because they're near and dear to us, right? So there was a couple really big news, or at least acquire acquisitions, right? So
Sure.
locally, there's a PRA Health Sciences. If they're a global organization, and there's a big site here in Kansas City, and they got purchased by Icon for $12 billion. John, wonderful. I think for Icon certainly gains a nice organization in PRA Health Sciences. I think for me, it's, I'm sort of torn. I'm happy for both organizations. I think it's a good fit, but also we're somewhat of a competitor. So I'm a little like, dang, right. But anyhow, it really is a good story. And I'm sure I'll be talking to some local friends and learning more about maybe the impact it's had. It's been a little too soon to
I,
see if there's
I,
any real impact.
yeah, I think the interest to me is the fact that last year, so this time last year, as things started to close down, first of all we were concerned about recession. Then it became obvious that pharma was the route out of the pandemic. So pharma has basically been booming, and we've been talking all the way through the year about the number of IPOs, and there have been acquisitions and mergers in the pharma industry, but the CRO industry has been relatively quiet. Even though the CRO industry has been benefiting from the work with pharma, we wouldn't have vaccines to the market this quickly without input of the CROs. And now it seems that we've got something to watch where in the course of it was just over a week, there's 4 different deals in
Yeah.
terms of purchases and acquisitions in CROs. So the big one, the huge one, is the Icon buying PRA, where we've gone from, I think, the fifth and sixth largest CROs by 2020 revenue to become second behind IQVIA, though they're still approximately half the size in terms of number of employees. Then you had Alta Sciences, who are a Canadian CRO slash CDMO, and they bought out WCCT Global, boosting its phase 1 and 2 offerings and also its geographic reach. And then they were bought by the European venture capital arm of Novo Nordisk. Then we had Wuxi bought these
Yeah.
two are
Gene
both
therapy.
very interesting. Two Chinese based or Chinese-owned CROs, Wuxi and Pharmaron, both bought units that would help them in the gene therapy space. So Wushi's ambition is to basically build a service so they become that one-stop shop for cell and gene therapy, and Pharmaron doing a similar thing where they want to have that ability to manufacture biologics, etc. So, like I say, it could be something to watch through the year. It could be the start of a trend, but to have 4 things happen so quickly, it makes you think. And Stephanie, you want to say something?
Yeah.
So I just wanted to say that with regards to where I see trends going, I think the cell and gene therapy support, especially in the bioanalytical community, is going to be a huge area of growth because from there's just not a lot of areas outside of this manufacturing CMC division where there is support for those types of projects externally. And as we get smaller companies jumping into that space, they're going to need bioanalytical partners to help
Yeah.
develop those drugs. And there just aren't very
Sure.
many options right now.
No, I think you nailed it, Stephanie. And these are all CMC related stories. So that or CR, yeah,
Mm-hmm.
however you want to think of it, manufacturing. And that's obviously, this is big news here. I feel like that always kind of leads into the bioanalytical end, so we're going to feel it more on the bioanalytical end.
Yes.
And I couldn't agree more. Like, for somebody who's been in the gene therapy space for 26 plus years now, I'm excited. We have things that even at KCS, some of the unmet needs that you're talking about in terms of bioanalytical support, specifically like flow cytometry, we've really expanded that. We've talked extensively about that on our podcast here. And then also things like ELISpot assays and then PCR. Those are the things we'll be bringing on in our new facility that we will be breaking ground on shortly. So couldn't say it better, Stephanie. Absolutely agree with you. I'm excited to see the future of drug development in the gene therapy space, and even and there's so much John just said around Alta Sciences being bought. We have a local Alta Sciences here, so that impacts us a little bit in Kansas City. But for the sake of time, let's move on to our main topic. I'm really excited to learn more about some of the main topic today is what COVID has taught us about limitations of the drug development process. We have a very special guest, as you all are getting to know Stephanie. We also have our Senior Director of LBA with us, John Pirro, and let's just jump right into our main portion. Why don't you, Stephanie, want to kick us off on limitations of current processes with increased data generation?
Sure. What we're seeing with the limitation of the current processes is really the pandemic has brought to light many of the issues that we had in the bioanalytical community where I live, but I'd say across the board in any sort of testing division. And I can say that explaining what I do now because of the issues and how it's become not only politicized but it's become news is amazing to me. I no longer have to go into great depth about what does testing mean because of just where we've lived in the last year since the pandemic really came across into all of our lives. So much of the limitation we're seeing is really, at least where I live, is in the data review process. We, what always, where I focus on is trying to provide software tools where it's a manual process. And many of the review that we're doing in the testing is by eyeballs. And I personally feel
Mhm.
that that is where you can lead to lack of efficiencies. Never mind the data quality issues you might have because we're all human. And I really do want to focus on turning those manual processes as much into uh, tool-based support that we can have, and not bringing the human out of it. I don't really aspire to, having robots replace all humans, but I do think that let's give the human the strategy part and understanding the context of whatever errors we might find, and have the routine and the exhaustive work of reviewing thousands and thousands of lines to a computer.
Yeah, Stephanie, I didn't set you up very well, did I? So
It's okay.
I think you finally got to the root of, like, sorry,
Sorry.
but like I want to recap a little bit of that and then maybe let John Pirro comment on it, just because I think
Sure.
we struggle with it at our organization. So as we have moved into COVID, and it's been well over 12, 13, 15 months now, uh, we've had to adopt the style of trying to take people off site more to allow for more social distancing. So there's a lot more QA, QC review being done virtually. And Stephanie, that's really where your software helps to with that. Do I have that right? Am I unpacking that correctly?
Yes, you've unpacked it
Yeah. And so,
beautifully. Sorry
John,
I stumbled.
John, maybe you share some of the struggles we've had at KCS, and I don't know if that's the right word, but maybe how we've overcome some of it and how this might play into it.
No, that's perfectly fine. Thank you, thank you, Dominic. And I just want to make sure that we were all under the same impression that we do not want to sacrifice quality at any aspect of what KCAS does. And in order to deal with that, especially with the COVID situation and how the distancing of laboratory space as well as distancing from the QC and QA groups, whereas before they were in here and dealing directly with our scientists, with the data going forward, is now that we have to have This distancing is now we have to focus on how we're going to quickly get all this data that we normally do from a visual point of view or a discussion point of view and get it all done. What's going on is the fact that now we've increased the number of days. So now, because of the virtuality of distancing, we now have 3 to 4 added or 3 to 6 days added on. So we get things back and forth from when we all scan it in and when we all discuss it and get back to the operations as a whole. Therefore, it was very important that the operational part of the Kcas bioanalysis, especially for an immunogenicity as well as for PK, adopt something that we can now add to our operational process, which does not interfere with the current QC and current QA quality aspect, but to see if we can adopt this process to see if we can find these issues earlier and faster. Therefore, when we start going into the virtual physical transfer of QC and QA, that we have now knocked out a whole bunch of things, and that the software that we're going to be talking about with Stephanie right now is now helping our operation to look at all these things before it goes into the QC and before it goes into QA, and we're not sacrificing quality and we're not sacrificing time. So Stephanie,
Well,
I would
that's
like
great,
you
John.
maybe just to give a little, okay?
Yeah, that was
So,
well said, John. And that's a nice maybe segue into Stephanie kind of jumping in and
can,
or sorry, John, did you have something? Go ahead, John.
can I just ask? I'd sort of ask a question. So, basically, what you're saying, because our next topic was how's COVID exacerbated the CRO process issues. So, basically, because we've had social distancing and the way we flow work, we're actually adding time to the time it time it takes us to complete tasks in terms of what from production of data to actually sending data to the client. Did I get that right?
That
Yes,
is
I
correct.
believe
Again,
so.
Yeah.
Yeah, so basically, that's a killer because we basically deadlines are critical for us. And we're always customers are always looking for how can we deliver things quicker. If we've got something that is automatically introducing a delay into our process, obviously
Yes.
we need a tool or a means that we can address that to bring our timelines back to where we really need them to be.
And
exactly.
also, and I'll speak to what I've seen out in the industry in general because this software tool came. I have a scientific advisory firm as well, and this came as a tool to our advisory, our my consultants out there, because of just the timelines that the review of the data that was overwhelming and was delaying, and much of that, um, the science would be moving along very quickly because many of the tools out there are either instrument facing, they're patient facing for HIPAA compliance. So everything is away from the review because
Hmm.
uh, that is where it's kind of a more exciting it's a more exciting process to enhance and to work on, whereas we're at the very end in a manual process trying to catch things that are moving quickly and more quickly towards us. And this was pre-pandemic. It was already an issue. We started developing this back in 2018, 2017 because we foresaw that there was an issue already, and that the science is where we want to focus. We want to make sure that we don't want to impede that, but we definitely need to give the reviewer enough time to make sure everything's done safely and appropriately. And that is where things were either being cut because timelines were crunched or things were delayed. And neither of those are the right answer to the situation, right? So we decided, well, maybe we should focus on providing a tool.
No, it's also important to understand that as the guidances get more detailed and we get more involved in the statistical from the immunogenicity point of view, it's very important that the operational consistency of performing these types of immunogenicity assays, ADAs, NAB assays, as we were just discussing, that the statistics becomes non-arbitrary. And it's very difficult to do that, again, with the COVID distancing and all this other stuff. And that's why taking on this software that was developed from an operational point of view is very important, because now we can look at that, put that into our operations, make sure that the process and what we're doing and delivering to our internal QC and internal QA is of a better situation, and then we reduce the overall time for QC and QA. So, Stephanie, if we could just
Yeah.
go a little bit more, I mean, do you agree
So
with
that's
that?
fine. I do agree with that. So there's two different core competencies that John's alluding to. We have the data review process, which was where we started off from, to help, the QC QA review process and bringing things uh, to being reviewed more in real time as opposed to at the very end of a study. There's also then we also saw a need for immunogenicity support because, as John mentioned, many of the current issues that we're finding are around setting of cut points, review of ADA data sets appropriately, setting sensitivity and understanding some of the more nuances around immunogenicity, and as you brought up earlier, um, the neutralizing antibody assays. And whenever you have a disconnect of an activity from the individual that's creating the data and the person that is consuming it, and in this situation, it's the bioanalytical scientists and the statistician, there was a general disconnect and this handoff where transparency was lost. And there is a big disconnect between understanding the biology of the situation and the stats. They don't always agree. And so, or they don't, it's not that they don't agree, they always don't understand each other's pain points and what each other's questions are answering. So just because something's statistically different doesn't mean that biologically it's different. And there was a lot of, as I call, scrubbing of the data because perhaps the statistician didn't understand that even though there is variability in the assay, that it was actually biological variability because humans are so different from each other, especially when you're dealing with a disease state like cancer, where the human's biochemistry is so altered, either because of different medications they received earlier or just their biological state in general. So, what we've done is in our system and in response to our clients' requests, is we've also then uh, developed an immunogenicity module where there's no longer, there's full transparency to bioanalytical scientists. We have statisticians involved in its production, and it's along with guidance, but it's all transparent and it's understood as well. And some of there's no ability to arbitrarily remove outliers. It's all within statistical analysis, and it also gives the user flexibility of perhaps inclusion or exclusion, of widening of criteria and narrowing of criteria, so they can really see the impact of some of their outlier tests on their data set and on their cut points. That was completely lost in many places. And so that we're really trying to provide insights back to the clients, our clients on the bioanalytical side.
Stephanie, you said so much there, and
I'm sorry.
I know, but it's great. I think it's great. It was really powerful what you were saying, and I think some of it might be a little above our audience, but that's okay. John, this is near and dear to you. I'd like you to maybe comment on some of that last piece, right? Especially where, in the immunogenicity space, it's an evolving field. It's probably in years to come going to continue to evolve. And Stephanie said some real poignant points around there's biology involved, there's some statistical analysis, and then outliers, right, John? Tell us a little bit. You want to comment on what she just described there?
Well, thank you, Dom. And thank you, Stephanie. I think it's a very important point to start thinking about from a bioanalysis point of view. And the reason for that is the immunogenicity that the CROs are generating have to be compared to the general public population. And we have to all understand that when we get these supposedly assays out into the bioanalysis field, it's very, very difficult because we're using a substituted type of positive control, which is not the actual antibody in humans, because you can't, it's impossible. So therefore, what we're doing is we're setting up and trying to validate an assay that's dealing with these from a population statistics and not a direct population. And that's important because now you start to have, okay, what do you consider as outliers? What do you consider as biological outliers and things like that? Because now you're dealing with such a broad population, and what you're doing in your laboratory is such a small percentage of that population, and that these statistics that are coming into play, specifically that are in the white papers that are out there in the industry, is showing that we have to be very careful of how we look at these outliers and look at the cut points and making sure that the decisions we're making and based on the results of our assays show that the public can probably benefit from this or not benefit from this, and that's the key point. So, by looking at an unarbitrary determination of outliers, as Stephanie was just describing, or the determination of these biological outliers, a system of non-arbitrary type of decisions have to be made. Now, again, we don't have that huge understanding of the statistics that are all out there, and we can't because obviously with COVID and everything else, we have these communications back and forth. Therefore, if we start looking at the AI software that we're looking at with Stephanie's group, we now have something that can be utilized within the operations to determine unarbitrarily if something is an outlier. And that can help us out because then if we can go back into the data, look at that, and then determine the cut point based upon an unarbitrary removal of these outliers, the data that we're presenting to the client and eventually to the areas of the government and stuff like that is going to be much, much better. So that's the goal, what we're looking at, and it's coming down to these types of decisions because, as you know, populations differ, and we cannot expand the current understanding of all the populations that we're doing in a CRO bioanalysis immunogenicity assay, and therefore what we're learning from Stephanie's teams as well as other people out there in the white papers is we have to start thinking very, very unarbitrarily and making sure that the statistics help us determine what to eliminate and what not to eliminate.
Thanks,
Thank you.
John. Yeah, thank you. Stephanie, you want to, I don't know, maybe we can move on to the next topic, or do you have any thoughts about what John was saying there?
So I do, but I also was wondering, Dom, would it be helpful to just address what is a cut point? In some ways, I feel like we might have jumped straight into the
Yeah,
discussion.
no, anywhere you want to go. We have plenty. We got a good, probably 30 minutes of time remaining. Or at least, yeah, go ahead. I think that would be
So,
great.
so I guess to back it up a little bit of what is a cut point and why do we care in many ways. Um, so I think of cut points is a threshold, and the biologics out there, and also this is becoming a big issue with some of the viral vectors used for gene therapy. Um, it is a threshold that's created statistically, as John said, by looking at a population, because the assays we're using and the population we're going after may have pre-existing antibodies already. If that antibodies are in response to, let's say, the viral vector that's used is sometimes just from the common cold. For example, someone might have seen something similar and they'll have an antibody and immune response. Or if you're dealing with a biologic, and maybe they've seen something similar in their cancer treatment. And so you really need to understand kind of threshold of the population and the general baseline of what a response would be if they have received absolutely no treatment. So you understand where does someone, what's their assay look like when they actually have no response, and therefore, when you're actually going into a clinical trial, where that threshold has been put is very important because then once they've received the drug, you want to make sure that they're not getting some sort of antibody response. I mean, there's the anaphylactic shock that happens, which is obvious from a clinical perspective, but there are other things that can happen later on that are perhaps not presented specifically in a severe cytokine storm, but maybe the drug stops working. And that's also, maybe due to ADA or immunogenicity response. So drawing that threshold appropriately is really key to understanding and treating your patient appropriately later on. So that's why it matters. And that's why it's important to do appropriately. And that's why understanding the variability of the population itself and not just removing everything so it looks really clean, that's where the danger lies if you don't truly understand your outlier tests and what you're removing from the population that you've tested before going into the clinic.
Yeah, it's the gene therapy piece is very critical,
Yes.
Stephanie. But let maybe talk to us. I think the next item was pain points around things, but or whatever agenda item you want to hop on next. But that was very good. Thank you for sharing that.
No problem. So general pain points in the industry, it goes back to the manual process. And not only trying to give, let's say, John's team some more transparency in understanding maybe if they're doing a cut point analysis, what they're actually trying to come out with in the end from the statistical analysis, but also removing any of these manual processes that are going to cause delay and are perhaps an even bigger issue when we move to a remote situation where you cannot be there in person because you have limited staff available in the laboratory. And we also are human. So humans make more mistakes perhaps if we appropriately set up the instrument or the software to run things for us. So removing those roadblocks in a manual process is really what we focus on. So anytime something is a manual system, we try to provide a tool to the human to either have better quality data going out. For example, when you're dealing with your clients, making sure that you've reviewed everything beforehand and that process isn't a roadblock in getting data out to your clients. And also just anything that involves transcription errors or anywhere that things can be misplaced or misconstrued.
Yeah, and I think that leads nicely. Tell us a little bit about how you're solving that. Right, that's the the importance of adaptation of artificial intelligence is what we're talking about here to regulators, right? So that's really what is the heart of what we call red thread solutions, right? Do I have that right? This is where your actual um, product can come in and really help solve those needs. Is that accurate?
Yes, it is accurate.
Yeah.
So
So
go ahead.
no, I was going to ask you to tell us a little bit about it, how it works, and take it wherever you want.
Sure. So the system was based off of the need, as I said, when I was reviewing data. We don't have what's typically called big data in our world, even though it feels like we're surrounded by a lot of it. But the big data where you can have true machine learning happening, that's definitely more on the clinical diagnostic side, perhaps, or other areas. However, we did need a tool to help review data um, and to revert that manual process and provide a tool to the reviewer. So what we've come up with is that its baseline is an expert system, a yes/no decision maker that goes along with guidance of the FDA and EMA and kind of best practices. So it's meant to be system agnostic, so that we don't want to force people to fit into our workflow. It could be laboratory informatics system independent, as well as we wanted to also fit into a variety of different formats that the data can come into. It could be a Word or Excel, so it's also meant to be agnostic to that. But it really would, it provides you upload your system, it's cloud-based, so it's also helpful for remote viewing as well, to be able to provide an insight into whether or not your data is meeting FDA and regulatory guidance in process, which right now that's not available to our current workflow. So that's what we've enabled to have. Like I
Excellent.
stumbled over that.
Yeah, no, you did. That's great. So, John Pirro, that is. I guess we got two Johns. Maybe I need to preface that, or Perkins, if you have something to add. But John, maybe you can just give a little bit of flavor of, um, maybe the benefits. I think we touched on it a little bit, but tell us your thoughts on some of this.
Sure. I mean, the key thing is that this is now becoming part of operations. So, in other words, what Stephanie was talking about is very, very important going forward with the quality of the data that we're trying to generate. And since we know that this is an arbitrary type of review of the data and looking at all this huge amount of data in a short period of time, that we can now quickly correct anything that's arbitrarily not looked at, and also correct it in operations. That doesn't mean we don't put it through QC and QA, but what it does is it sort of hastens and quickly gets through QC and QA now, because operations is looking at this data from a quality point of view. So that's basically
Yeah,
the power of what we're looking at.
yeah.
And
It's. Oh, go ahead, Stephanie. Sorry, go ahead.
well, and also how we came about it is by mapping out data over and over again. For as I said, with the scientific advisory firm that I have, we have data from 7 of the top 10 contract labs that are out there creating these data sets. And we just mapped a thought process out from looking at actual raw data in Excel, Word, and PDF, and created decision trees over and over again. And really, that is where not only was it me mapping out my thought process through data set, but it's also the people that worked for me, the consultants, as well as my QC staff that's at Red Thread Ariadne Software itself. So by doing that over years and years and training the system to find nuances in a data set that perhaps I as an individual wouldn't have found, but my teammate would have found, and that's the power as well. So
Yeah.
it's a force multiplier, because it's not just one reviewer. It's almost like 40 reviewers are looking at your data set. And also, by having it actually, that was actually not the hardest part. The hardest part was the parsing of the data, making the system smart enough to that the user input is at a minimum. Because if the user has to input all of this data, all of these upfront, then there's gonna be a barrier to entry of use, because why would I want to do that? I might as well just look at it by hand then, or write a macro. And so we did make it smart enough and build up its own dictionary to understand the difference in nuances in how maybe CRO A labels their samples versus CRO B. And by the training over over years and years, not only that parsing, so the system's smart enough to know what checks should be, how it should look at the data, and then the checks themselves. So that's really where we've saved a lot of time in the last few years of just training it, and the diversity of the data sets that we've provided it.
Yeah, that's great. So it sounds like you have a good way to get the information into the system, and then, um, we're not taking away any QC/QA review. Like, that's a
No.
critical piece of this, but get it into a system that allows it to have like base. I heard you say like 40 sets of eyes. That's so powerful, right? Like, done
Mm-hmm.
a lot of QA review in my time, and even when you manage a team of people, they all QA a little bit differently or QC differently, and some,
Exactly.
it's not to say that not everybody's got quality about them, but it's, you fall into some maybe patterns, or you start looking at different, just the eye can do things to you, and to have it be able to amplify 40 people, that's a very, very powerful portion of this. And really, it's and you really touched on, hey, we're not going to make it hard to do this, right? We don't want to add
No.
another layer of complexity. We want to just make a better quality product at really, without actually losing any efficiency. So really good stuff.
And it's simple flags as well. I liken it. We instruct the reviewer of where there may be issues, but the context really is reliant on the human understanding the situation. We have simple alerts of yellow, green, and red with some text following afterwards to point the reviewer in the direction of where they may need to go. But the person needs to understand the context. But having lived in this world of reviewing data in various different ways, let's say if you had 8 hours, I was spending 7 of my hours finding the problems and 1 hour solving. And we really want to flip that around. We should have 1 hour finding the problems with the tool assisting us, and then 7 hours solving the problem, or let's face it, doing something else because everything is flying at us so
Yeah.
much quicker than it ever was before.
Yeah, that's well said. You're right. You spend a lot of time trying to find a problem, and fixing it is really not usually more than but a few minutes. Yeah, no, but you're right. The energy is what you're also referring to in terms of. I talk a lot about energy and focus these days, and that would
Hmm.
be a big part of what you're able to conserve on, and then focus on other things. So really great stuff.
Yeah,
But go ahead,
that
John.
that's what I'm saying too. It's like based on that, it really means you use your people in the more valuable part of the process rather than, like you say, the trying to find the issue, and yeah, it's that's a big deal.
Yeah, so this has been great. I think we covered all of our points here. So we've got Stephanie. You're not off the hook yet. You are going to have to get on the hot seat. That's just traditional. But maybe some parting thoughts from. Let's go. If John Pirro, maybe you can go first, and then Stephanie next. Just some last thoughts on this topic. And it's been we're not quite done yet, but it's been a lot of fun.
I just want to let you all know this. What Stephanie is saying is very, very true to the operation. The team that's been using this is so excited because it doesn't add more time to their time, but it does give them a better understanding of what they're seeing because they've been seeing and looking at these reports and data every single day for days on end and generate them so they can miss things. And by having this come back and say real quickly, and we're not talking a long period of time, just pressing a button and asking them to look at this, finding these things as red, yellow, and green helps them tremendously. So it's a really, really good benefit to the operations moving forward. That's all.
And Stephanie, thanks, John. It doesn't surprise me at all that this is a lot of people are excited about it at KCAS, and I love the idea of having just again things that can that red, yellow, green concept's great, and quickly the power is the quickness of it is also it. So Stephanie, any parting words before we put you on the hot seat?
No, I guess to go kind of talking to concerns around the AI and regulators. The regulators are looking for also AI tools, in just in general across the board in drug development to help with efficiencies and to get, our main goal right at the very end is to get medicines to the patients faster. And really, that's what this helps does. It's an area of inefficiency that we're trying to add a tool to help the scientists move things forward, not remove the scientists. So there's a monetary benefit for the companies as well as a human unmet medical need benefit as well.
Yeah, I think there's a whole nother topic on agencies and what you just said there, but I love it. I think it's great that you're starting to really. This is a forefront to me, and an area that I'm sure the FDA is going to welcome, but keep an eye on as well in terms of
Mm-hmm.
like just the regulated. It's making my mind hurt a little bit about how you could actually get this, like maybe not necessarily. I don't think you need like I think of like 510(k) clearances, but whatever the sort of clearance you might need, or maybe you don't. But either way, I think it's something that would certainly be embraced with any of the meetings that I've been at. You nailed it. The FDA is always looking for ways of improvement, and this certainly meets an unmet need. So
Sure.
well done. Um,
Can I
yes,
ask John Pirro
sure.
one question? So, John, obviously you've been involved in the implementation at KCS. Where in what group is it currently being used, and what's the plan beyond that?
Sure. The current group is that's using it is the large molecule LBA regulatory group, which has the PK for large molecules as well as the ADA immunogenicity. So it's being done there first, looking at it, and then obviously later on, we'll probably look at it at other parts of the corporation right now. Does that answer your question, John?
Sure, I think so. I mean, so you're mainly, yeah, yeah. I think so. What have you done so far with it? And then, yeah, I'm just interested as to how you're implementing it at the moment, and then like say where you see it going with it within our environment.
Well, Right now, we have a couple of PIs that are currently using it, learning it, and making sure that everybody understands the run. We're practically using it for everything we're doing currently. So obviously, we started, a few
Okay.
people, they enjoyed it, they thought that the ability was fantastic, and then we incorporated it to every PI now within the large molecule corporate group.
Yeah, is John? It's regular, like anything that's a GLP study. Are there plans to move it to non-GLP, or will this be mostly in a GLP space? And maybe that's to be determined. Maybe I'll help
Uh,
you answer that question.
there you go. But we are using it for GLP studies as well. Yes.
Oh, so to that question, what we have so far as modules are concerned for Red Thread is that we have large and small molecule PK modules that can review method validation and sample analysis data sets. And we right now are working with John and his ligand binding assay group, but we do have small molecule support as well. And then we have the immunogenicity modules that do the validation sample analysis checks as well, but then also the cut point analysis that's ongoing, and that's been the big thing that we've helped out with, as well as the PK validation and sample analysis checks. We have a
Okay.
report populator as well that populates the report. Since we're already looking at the data tables, we're populating it into the spot where it goes. So it's not a writer, but it at least puts things. It's reviewing. It's taking away that manual process, as you can see where I have a tendency to go. Is anything that's manual that I can farm
Yeah.
out to a computer? Why not?
Yeah.
But
No.
the
So
yeah.
it sounds like it's PK, ADA. No bio, like it could be probably adapted to biomarker, but I'm going, I want to know about multiplexes. I got 1,000 questions. Can you do it for flow cytometry? Blah, blah, blah. No, but
Well, that's actually what we're moving on to next. We actually are writing a large SBIR grant to focus on because we can do multiple analytes at once right now with our system, but everything's hard coded to GLP regulated clinical
okay.
bioanalytical
Okay.
criteria. But we are moving
Yeah.
on that.
Oh, that's great. And it sounds like this is an area, this is going to be a growing area, Stephanie. I think there's a lot of interest in what
We're
you've
moving into flow,
discussed today.
so we may want to talk separate, you and I, because
Yeah.
we're moving
Yeah.
into flow
And the
next.
first thing would be like, a 10-plex or something, and then
Yep.
maybe flow. But it's just so manual right now, with some of the processes and not so manual, but like just in terms of the quality of the data and all of that gets to be
Mm-hmm.
much higher if you have something that can flag things as you're describing. So just great stuff.
Mm-hmm.
And I don't have any other questions for you. I think it's been a wonderful opportunity to spend some time with you, learn more about this exciting product. Um, and maybe with that, you are going to get in what we call our hot seat. And trust me, it's not that hot. The first question is, what is your favorite episode of our lovely KCS podcast?
Okay, great. So a few weeks back, I forget, although I feel like I'm living in dog years, right?
Yeah.
But I believe John did one just previously on biomarkers. Am I correct? We had a big, nice
Yes.
discussion on that was a really good one. But I did like the discussion. I love your news section. I find that extremely informative uh, because it's nice to have a snapshot of what's going on in the world, especially when it comes to vaccines and how things are developing. But even like the news you talked about today about the PRA being purchased by Icon. Sometimes we're highly focused on our work and we don't look at the world around us. But I am disappointed that you didn't bring up Dolly Parton and her vaccine to Jolene. That was an amazing bit of news this week.
I guess I missed that. So, if my wife were
Oh,
here, she'd start singing "Chlorine, Chlorine." She likes to do a parody.
exactly.
So, tell us about what I don't know if I knew about
Well,
this. Tell us about Dolly.
sh-
Oh,
she,
well,
yeah, I don't want to talk too much, but didn't she invested in, was it the Moderna vaccine?
she did invest in the Moderna vaccine, and I'm a big
Yeah.
Dolly Parton fan anyway, but then I love her even more because she sang to the tune while she got videotaped being vaccinated and sang to the Jolene vaccine throughout the whole
Oh, I
process.
missed that. Oh, that's great.
Oh
It's
man,
really
I can't.
lovely. You
I
have
mean,
to search
I have
for
to.
it.
I have to say, I mean, Dolly Parton. I mean, it's just fantastic. The whole book
Hmm.
initiative that she does in terms of supplying books for kids in terms of reading and
Yeah.
stuff. I mean, I think she's great in all that stuff she does, and I can highly recommend Dollywood as a destination too. I did that
Yeah.
many years ago with my wife.
I
So
haven't
so
been
it's
there, but yeah.
yeah. I have a little sidebar. When we were in LA a couple years ago, I think it's the last vacation I took. Um, at one of the museums we were visiting, there was I think it was the Grammys, and um, the exhibit was on Dolly Parton and Johnny Cash. So I got to learn a lot about Dolly. Saw a lot of her. She was very petite, right? A lot of her clothing was exhibited, and a whole big exhibit on her. My wife was. I like her, but my wife's a really big fan. So I will find this video and show it
Yeah.
to her, which is great. You heard me talking about chlorine. She don't. If you hear the song Jolene, she's going to start singing chlorine. She's got her own parody, but. Um, we're gonna wrap up our heart seat here with a couple more quick questions for you because we do love to talk about food on this show. So if you haven't figured it out, still haven't cracked open my new Uni pizza oven yet, but I tell you it might be coming because the weather's warming up. But what's your favorite dish to make?
Okay, so if I wasn't going to be a chemist, I would have been a chef because I do love to cook. So my favorite dish to make is actually crepe Suzette.
I heard crepes, and then what type of crepe?
Suzette.
Crepe
Oh,
crepe Suzette. It's
that
really
that
good. It's
that
really
sounds.
good.
So do you make your own? I take you make it all handmade, right, from
Yes,
scratch
I
and
do
everything.
from scratch. But I also learned how to make bagels during, everyone had their bread craze during the initial COVID, and mine was bagels. And
Wow.
New York style bagels, because moving from Philly back to, to Kansas last year, I missed my New York style bagels. So I learned how to make those as well.
You
So,
are
so next time I come to Kansas, I know where I'm staying.
For
yeah.
sure.
Forget the hotel.
I think
We have a big,
the next time we invite you to KCAS, you're going to have to bring us some bagels, I guess. But that is fascinating.
I,
You're the first person I've ever heard say they're making homemade bagels. That's great.
yep. It's a 2-day process, but it's really good.
Yeah, my pasta sauce can take me 4 days at times. Just depends on how big
Hmm.
a batch. No,
Exactly.
no, that's great. I'm trying to envision how you make a homemade bagel, but I don't think we have time for you to educate my somewhat less. I'm not a big baker, so but I love to
Mhm.
cook, and it's fascinating that you've touched on some really unique items, in my opinion, that you don't hear a lot of people talking about making their own crepes and their own bagels. So
so actually, John's our guest too. So, John, are you gonna? Can you answer this?
Well, I started I started to make some of my grandmother's recipes for Italian. I found a delicatessen in Kansas City, and I'm making ricotta, using the ricotta into making some nice lasagna and going forward with that. But that's what I've been doing.
oh, John, we're gonna
Me
have to
too.
trade. There's a barter system going on here. I'm gonna have to give you some sauce. I want some regatta. Can't find good regatta. I missed that. That sounds fabulous. I like making stuffed shells with mine rather than the lasagna. But either way, I'm getting hungry as we sit here.
Okay, so I'll bring my
Um,
bagels and you all make lasagna. It'll be good.
There you go. We'll all eat together.
Gain 20 more pounds.
There you go.
Yes.
maybe one last question, John. Why don't you
Sure.
take us away with the last one?
Well, we could. Well, the two shouldn't take long. So, what's your favorite adult beverage?
Coffee. um, if it's not a caffeinated drink, then I guess it would have to be a good glass of champagne because
Oh,
I am that pretentious.
no,
That's
that
not pretentious at all.
not at all.
A
A
good
lot of
a good Veuve Clicquot is my favorite celebration drink.
no. That's. I think coffee was acceptable too. How about you, John Pirro? I might even know. Maybe I can guess yours, but why don't you tell us?
I like macchiato in the morning. That always sets me off. That gets me going. And then there, and as you know, I like a vodka martini, very cold with a slice of lemon in it.
Yep, little lemon
Sh-
spritz on there.
sh-
I do know.
yeah, is it shaken or stirred?
Oh,
Shaken, of
Shaken,
course.
yeah. John, we also have a palate for our whiskeys, but we're running out of time now, aren't we?
Yes,
So
I know.
Oh, actually, on just very quickly on whiskies, we had fantastic news today, in that the US is talking, putting on hold the last year there was talk of raising tariffs on Scotch whiskey by a huge amount. It was like pretty much going to double the cost of whiskey. That has been phased out, so we're back to hopefully reasonable whiskey prices. I'm a happy camper.
yeah,
As
that
That makes me happy. I'm going for that.
yeah, that's the best news I've heard on like taxes going down, and you just made my Thursday because
You got that right.
yes.
uh,
So so last but not least, what are your weekend plans?
gardening.
Okay.
Gardening outside. We have moving back here. We ended up getting 6 acres in the middle of town of Lawrence, which is crazy, right? And we have an area out back. I grew up in a desert, so my husband is the gardener, and he has this big old plot of land. He wants to plant a whole bunch of tomatoes, things for me to cook with. So he's going to garden, and I'll do the cooking. So yeah, so we're going to spend this weekend to get it all prepped to plant our vegetables.
Yeah, our backyard's still under snow, so we're just sort of discussing what we want to grow this year. We're not anywhere near ready to get going yet.
I'm very jealous. I live in an area where they do not let me have a garden. So in the
Oh
last
no.
couple of years, I
Really?
have not. Yeah, it's frowned upon. I mean, maybe I don't even know. I think there's so many crazy bylaws and stuff around. We have like neighborhood homeowners associations. This was new to me after I had moved from the East Coast. They have a lot more control. You certainly can never hang your clothes out, which again
Sure.
seems a little strange. But yeah, I don't. You know, you just it's just I think it's heavily frowned upon. Um, and I miss I had a garden at my own home, and
Yeah.
I might have to come poach some from Stephanie, but
That's
I'm like hardcore.
absolutely fine. You can come
I
and
had
I'll show
like
you how to make bagels. I'll show you how to make bagels and you can have some tomatoes.
well, I'll certainly barter with you. If I
Very
had
good.
like 15 tomato plants, and at one point I had as many as like 40 when I lived in Pittsburgh. And I used to, anyhow, I would can a lot of tomatoes and salsa and even just tomato juice and pasta sauce and paid by
Hmm.
parking at the school with tomatoes. I'm a bartering guy. I'm old school like that. No, John Pirro, tell us if you have any exciting plans with the weather warming up here in Kansas City.
Well, actually, I'm going to be spending more time inside my apartment. I'm making for the first time a ricotta cheesecake, so I'm working on that going forward. But then on Sunday, I probably will go out and travel west and just get love the atmosphere and just be exposed to the open spaces
Yeah.
of Kansas because I've been in New York for so long. It's nice to see open spaces.
Yeah, and if you hit to Lawrence, you can. There's plenty of breweries and nice eateries on the way west, and I think you can even track where they sell some maps where you might want to stop and see some little destinations or at least good places to get fried chicken.
Good.
Right, that's popular
Yep.
Yes,
around here.
If
yes.
you do make it to Lawrence, John, yes, let us know. We're right here. But there are some great breweries here, including Lawrence Beer Company is wonderful
Yeah,
Excellent.
out of Lawrence.
that's one of my favorites. Yeah, and John, the ricotta cheese pie. I haven't seen one of those. I don't even think they have it at my little Italian mart. I like to go to market that I like to go to, but I'm certainly. I ran out of some spices and stuff. So I'll be going down to our what's called our River Mart downtown. And if you haven't been down there, it's great. But I'll look. I want to look for one. I love a regatta cheese pie, John. I
I
think
know.
so. I'm now like salivating, and I don't know. I maybe have 10 quarts of pasta sauce left over, and I might be doling out 2 or 3 of them to the chagrin of my family in order to barter some stuff. So that was lovely, guys. John Perkins, why don't you tell us your weekend and maybe I'll close this out.
Sure, yeah, my weekend. So I don't know if you've heard any noise in the background, but we actually have we're upgrading our bathroom after being in this house for 23. Our main bathroom after being in the house for 23 years. So that's going on at the moment. So there's been a lot of drilling and sawing and banging. But one of the biggest issues over the weekend, because the guys won't be here, is that the two young cats have figured out that if they can get into the bathroom, they can get into the wall and disappear off into the roof space. So we're going to be probably spend most of our time guarding the bathroom doors to keep them out. Apart from that, it's still snowy here, so it's wash the car to get the salt off it. Just a general relaxing weekend, but not a lot planned. Like I said, big news is next Wednesday. That's the big deal for me.
Yeah, exciting. I have more lacrosse is taking up and consuming my weekends. My son, who's a freshman, is made a lacrosse team as a JV player, and we're last weekend we spent 6 hours outside. We caught a break. It was a nice day, and then we'll be watching some more lacrosse tournaments. He had a jamboree last week, and then he's got a couple games this weekend. So very excited to spend some time outside, even if it is maybe in a lawn chair. But either way, enjoy that this weekend. And with some luck, I might be able to pull out my oven. But and for me, I'm actually maybe going to be able to take a small vacation, John, and that will be exciting as well. But maybe the next time we have a podcast, I'll share what that'll be about in terms of. I say maybe because it seems like forever since we've had a vacation, but anyhow,
very much deserved.
Yeah. And with that, I want to thank our guests. Thank you both, John Pirro and Stephanie Pasas-Farmer, for their time today. Any parting thoughts before we let y'all off the hot seat?
I'm fine. Thank you very much for the opportunity to discuss this.
Thank you, and I hope you all get the chance to see Dolly vaccine.